Here I will discuss eukaryotic transcription and some of the factors that play a role in the process. I have here the coding strand going from 5' to 3' in black and the template strand going from 3' to 5' in white. On the strands there are various recognition sites for transcription factors and other proteins to bind and regulate transcription, with one of the most important ones being the Tata box within the core promoter. On each side of the Tata box going upstream and downstream, there are B recognition elements and also an initiator site which has the +1 site which RNA polymerase II binds. For transcription to occur, the pre-initiation complex must assemble correctly. First, the Tata binding protein with TF2D needs to bind to the Tata box, and TF2D is the only transcription factor that makes a sequence-specific contact with the DNA strand. TF2A is the second transcription factor to bind, and it binds to TF2D and the Tata binding protein area, stabilizing the interaction between TF2D and DNA. Once TF2A and TF2D make contact with each other, this is a protein-protein interaction. TF2B is the third transcription factor to bind, and it binds to the B recognition element and is critical for absolute stabilization of the TF2D DNA complex. TF2F binds to RNA polymerase II, which is dephosphorylated at this point, and recruits it to the pre-initiation complex with the help of TF2B. TF2E is recruited to the pre-initiation complex with the help of TF2F. TF2H and mediator complex are also recruited to the pre-initiation complex after TF2E. TF2E is required for promoter melting and the recruitment of the kinase active transcription factor TF2H, which acts as a helicase and unwinds the DNA strand to achieve an open complex. So we're going from a closed complex to an open complex. RNA polymerase II recognizes the +1 site and binds to it, and initiation can begin. The first phase of initiation is when approximately 5 nucleotides are synthesized and then excised out due to instability. This is called abortive initiation. The second part of initiation is promoter escape. This occurs when polymerase II escapes from the promoter region, having synthesized about 10 nucleotides that escape over abortive initiation, that favor escape over abortive initiation. Productive initiation is the third step of initiation and occurs when approximately 25 nucleotides have been synthesized and elongation is signaled through the 5' end capping event in recognition of the phosphorylated C-terminal domain of the RNA polymerase by the capping enzyme. The capping enzyme adds a 5' cap which protects the 5' end of the nascent mRNA strand from exonucleases. mRNA is then cleaved and polyadenylated on the 3' end at the poly A site. Once elongation is signaled, transcription factors leave the RNA polymerase II and the DNA strand, and RNA polymerase II is dephosphorylated and can then form within a new pre-initiation complex.